Here's a quick overview of meiosis, slightly different way of looking at meiosis, and so we're going to follow one gene and its two alleles during the events of meiosis. So let's start off with maybe some chromosomes. So here's a pair of homologous chromosomes, and so this could be chromosome 1 from mom, and this could be chromosome 1 from dad. That's totally fine. So we're just going to follow one pair of chromosomes, otherwise it gets incredibly complicated. And so what I'm going to do is I'm going to put a gene on here, and we're going to go back to our old friend, the T allele, the T gene with its two alleles, big T and little t. So before we go into actual meiosis, the first thing that's going to happen is that DNA replication will occur. And so if we replicate these chromosomes, then we replicate these alleles. And now these are still a pair of homologous chromosomes, except we've got replicated sister chromatids. So the first thing that's going to happen is that in meiosis 1, we're going to line these, all our homologous chromosomes up in a cell. So keep that diagram in mind, and let's just think about all of the chromosomes in a cell. And I'm going to think about an organism with 3 chromosomes, just for the sake of simplicity. So let's redraw this. Here is our pair of homologous chromosomes that we started out with. Let's just put 3 other chromosomes in here, just so we can track them. So here are 3 other sets of replicated chromosomes. So remember, what meiosis is going to do is it's going to pull the homologous chromosomes apart to opposite ends of the cell. And so if we think about what's going to happen there, as we separate those two sets of chromosomes, let's move them to opposite ends of the cell and just erase these ones in the middle. And so when they get separated out, so great. That's the first meiosis. So that's meiosis 1. So that's what's going to happen in meiosis 1. Let's now take all this stuff away and think about meiosis 2. So that's meiosis 1, and I'll just draw the cells that would be produced, and then we're going to think about meiosis 2. So from meiosis 1, we're going to get 2 cells. Both would be haploid, so we'll just mark them as haploid by saying they're N. That just means there's one copy or one set of each chromosomes. And so in this one, let's put that homologous chromosome, and then we'll draw the other 3 in just for completion. Okay, there's the chromosome we're interested in, and here are the other chromosomes. So that would be meiosis 1. Now let's think about what happens in meiosis 2. In meiosis 2, we're just going to think about what happens in this top cell, and the same is going to be true for the bottom cell as well. So in meiosis 2, the replicated sister chromatids are going to line up like they did before in the middle of the cell on the metaphase plate. So we're only interested in this top cell right now, but the same thing's going to happen with the bottom cell. And what we're going to do here is we're going to pull the sisters apart, and so we're going to pull those into 2 different cells. And so what will happen there is we'll end up with, again, 2 cells, and this cell will have that allele, this cell will have that allele, and then we'll have our other chromosomes that we have to worry about. And if you do the same set of events with the other cell, with this cell over here, you're going to get exactly the same thing, except the alleles would be little t. And so you'd end up with 4 cells, each cell with 1 allele. And the allele can either be big T or little t. So that's another way of visualizing meiosis.